I'm Jack Crawford. It's a pleasure to be here. I'm the CEO of Demeetra. And thank you all for being here, and thank you for giving me this great opportunity to present our platforms, our technologies to you. I'm going to talk about engineering CleanCut CHO platforms from CLD to GMP with continuity, and thank you. All right, so at Demeetra, we are solely focused on trying to solve our clients' biggest, most painful problems. And the first problem that we really attack is the fragmented CRISPR IP landscape, which creates very large uncertainty. There's probably 5,000 patents on the Cas9 protein. And there's a lot of companies sitting on the sideline with this great technology that can be used to target genes, improve your hosts, deliver genes of interest. So we find that this is a big problem we're trying to solve, and we have a solution. We call it Cas-CLOVER. I'll talk about it in detail. It provides a clear freedom to operate path for commercial cell line development. Another problem that we try to solve is many genetically engineered CHO lines are captive to service contracts. So that means in order to get access to a specific genetically engineered CHO cell line, you need to lock in with that service provider. Otherwise, you can't get access to it. Or sometimes they actually, either a service or a cell line platform will carry a royalty on the molecule itself, which significantly reduces the value of that molecule. And if you're a startup, it reduces the value of your entire company when you're looking to partner or sell that asset or that company. So we sell our platforms directly off the shelf as an option, and then we also package them into a whole workflow. And now I'm very excited to talk about today that we have established a partnership with the downstream CDMO where we're now eliminating the CLD to GMP resets through a seamless single contract path. So you get one master service agreement, and we can take your gene of interest all the way to clinical material, but all the way to commercial material as well. Demeetra is based in Lexington, Kentucky, where we're known for bourbon and horses. But we also have an incredibly expert staff of scientists. And we work with these scientists work with our technology every day, always trying to improve it for our clients and developing and validating protocols. And then we focus on transferring those protocols, transferring that know-how to our clients. And one of the strengths that we have from being in Kentucky is we have a significantly lower overhead than coastal CROs. And therefore, we can provide services, but also our technologies, we can provide and pass on that savings to our clients. So to go over our cell line development platforms as a, is there a pointer on this? No. Okay. To go over our cell line development platforms, starting with Cas-CLOVER. Cas-CLOVER is a targeted nuclease. It's a dimeric nuclease, and I'll talk about its mechanism and its function in detail, but it provides very high efficiency and has improved precision versus CRISPR-Cas9 because of its dimeric nature. That's a well-established advantage versus CRISPR-Cas9. It's been published many times. It also creates larger deletions versus CRISPR-Cas9, and that results in more complete knockouts, and it has a staggered cut when it cuts versus a blunt-ended cut, which I'll show you later, which results in higher targeted integration efficiency. We have a completely license-free transposase platform that we call Harbor-in transposase. You buy the reagent from us off the shelf, you pay the reagent cost, and then you're done. There's no license to negotiate, no milestone payments, no royalties. It behaves a lot like other transposases, where it integrates stably into highly expressed sites for driving high productivity. It also has a large transgene cargo capacity, so that can support complex molecules, which were presented earlier. It can also support huge capacities. We've done 200 kb. We've inserted, I think, 10 or 12 genes all at once in a single cassette. And then we're using Cas-CLOVER to build a platform of suspension CHO-K1 cells, edited platform, which we call the CleanCut CHO platform. So these are engineered host cells. We're expanding this collection using Cas-CLOVER, and we're focused and we're targeting improvements in selection stringency, productivity, and quality, product quality. So first, to talk about Cas-CLOVER a little bit versus CRISPR-Cas9, you can see Cas-CLOVER is on the left, and it's this dimeric system where you need both the left and the right guide RNA. The guide RNA is complex with a completely nuclease inactive Cas9. So it's a dCas9 protein. And so it binds to the target site, but does not cut the gene at all. And then instead, it's fused to a dimeric nuclease called CLO51. So by dimeric, I mean that there's no activity unless both the left and the right come together at the target site. That's what reduces the off-target effects that CRISPR-Cas9 has. On the other hand, you see CRISPR-Cas9, it's a monomeric enzyme. You get the guide RNA complexing with an active Cas9 protein that binds to the target but also cuts it. And then if it binds by mistake elsewhere, it will also cut that. I also want to mention that there's a flexible spacer region in between the left and the right guide RNAs for Cas-CLOVER, which allows for targeting of any gene in any genome. Now, I mentioned the freedom to operate on our system, and I want to just briefly go over that and how we can claim that. We have licenses, or we've developed our own IP on every single component of Cas-CLOVER. So with the guide RNAs and the completely nuclease inactive Cas9 protein, we have a license from ERS Genomics, which is the licensor from the CVC, California Vienna Charpentier. This is Jennifer Doudna and her groups. They have all the IP that you need for a dCas9. So we have that. Then we have an exclusive license from both the Helmholtz Munich, which invented CLO51, and Poseida Therapeutics, which developed this whole system. So we package that all together and we give that out as a license package, or however we want to structure it to our clients. Cas-CLOVER cuts differently than CRISPR-Cas9. You can see the two guide RNAs shown here, the left and the right, and then the PAM sites. Cas-CLOVER always cuts in between the two guide RNAs, and it creates this staggered 4-base overhang cut. And that's what we believe drives that higher targeted integration efficiency, higher knock-in efficiency. It also reduces translocations. CRISPR-Cas9 has a lot of translocations associated with it, which can destabilize your cells. Cas9 creates this blunt-ended cut, and so those are the differences there. Now, getting into some data with Cas-CLOVER, we recently published this data in Biotechnology Progress, where we were looking at the efficiency, the editing efficiency of GS targets in suspension CHO-K1 cells. So there are two GS genes. One is the GS5, the one on the chromosome 5. This is the primary gene that people have been talking about for quite some time. And then there's a newly identified GS1 on chromosome 1. the pseudogene. And there's data published that if you knock out that GS1 as well, you'll have higher selection stringency. So we wanted to target both of them. And so we were looking at the editing efficiency at first, and you can see the black bar, we have both the left and the right guide RNAs with Cas-CLOVER, and we get very good editing efficiency in both targets, around 80%. And then with just one of the guides, the left or the right with Cas9, you still get very good editing efficiency with CRISPR-Cas9, slightly lower. Now, we also have a recent publication from one of our partners, Sanofi, who published in Biotech Progress recently, showing enterprise-scale host cell engineering with Cas-CLOVER. So this is the design on how they knocked out their GS. And you can see in the red bars there, the 2 guide RNAs and that 27 base pair spacer region that they had. They targeted the intron-exon border at exon 5, and there's a active amino acid that they were targeting. But they have a large collection, in this case, 30 different GS CHO hosts, all with different characteristics that they wanted to exploit, and they wanted to make those GS knockouts. So they had 30 unique host cell lines, created nearly 100 GS knockout clones, were generated in 6 weeks with Cas-CLOVER. Another one of our partners a couple years ago published, in this case Takeda, published the use of Cas-CLOVER for editing HEK293 cells. And I put this in here because of the complete knockout. Again, they had very, very high editing efficiency where they went right into single cell cloning. For this target, they didn't have a selection. And they had 13 clones, most of which were knocked out. Only one showed wild type alleles. So they had 92.3% knockout efficiency. They looked at the indel, or the deletion length, for these clones and saw what we would expect, larger deletions. You can see most of them are around 20 base pairs up to 50 base pairs, but some of their clones had very large deletions, over 150 base pairs. And these knockouts resulted in complete protein knockout. So they were looking at STAT1, and you can see the protein is completely gone, other than that one clone that still had the wild type knockouts. So here's a little bit of our knock-in or targeted integration data, where the experiment we did was we wanted to test the system without selection. And so a pretty simple way to do that would be a promoterless system with your arms of homology, which are shown there in the blue, surrounding GFP, which is promoterless. And we were targeting GAPDH with this system. And then we also wanted to track the transient expression. So we had a RFP, or TD tomato protein, on the backbone of this vector. So we could track that. And we got what we would expect here. We're showing the cell population and the different types of cells that are being expressed over time. And right at transfection, we had a very high population of transient expression that you would expect, and low population of our targeted knock-in. But over time, at about 2 weeks, that transient expression goes to 0, basically, and then the targeted knock-in expression goes way up to about 20%. So this is a very high efficiency strategy for knocking in your gene of interest in a safe harbor site, or construction of landing pads for recombinase and integrase technologies. So now getting into making our cell lines. The first cell line, we call it the CleanCut GS CHO cell, where we adopted a CHO-K1 to suspension culture, and then we knocked out the GS. I'm going to show the details on this, where there are the 2 GS genes. And remember, we need 2 guide RNAs to target a gene. And at chromosome 5, one of our pairs wasn't going to work. And we did this specifically to try to see if we could target one versus the other. And so the pair in red is targeting exon 5. So that one works, makes its deletions. The pair shown in blue is in the chromosome 5 gene, there's an intron. So it's too far away from the other guide RNA to have any activity. So it was inactive. However, on chromosome 1, there is no intron, and therefore both the left and the right guide RNAs are close enough to have that activity. We get that cutting on chromosome 1. But the red guide pair had SNPs, had some mutations, and it wasn't active. And the reason we wanted to show this is a really nice system for targeting or for discriminating between highly homologous genes within the genome if there's ever any interest or of doing that. So this is showing just the deletions that we got with this CHO knockout, which is what we would expect. You can see the 2 guide RNAs, and then 10, 20, 30 base pair deletions in our edited clones. The first thing we wanted to do was look at glutamine sensitivity, and you can see it's very, very sensitive. At day 9, most of the clones are completely dead without glutamine supplementation. And then we wanted to see if we transfect these with our Harborin license-free transposase, do they produce antibodies? So we used trastuzumab to show this expression, and we do not have any AMBER systems or any bioreactors. Instead, we just rely on fed batch 24 deep well plates and tube spins, and you can see we have 7 grams per liter and over 100 picograms per cell per day using this cell platform with Harborin transposase. So now introducing for the first time our next CleanCut cell line that we're launching today. It's the CleanCut GS ADCC plus cell. It's another obvious target, and some of you guys are aware of it, but one of the reasons you would make this is antibodies that have fucose do not have as high of effector cell function or antibody-directed cellular cytotoxicity. So when you get rid of that fucose, you make afucosylated antibodies, it can interact with the effector cell better and result in higher potency, higher cell death. So this is extremely important for oncology, but also autoimmune, inflammatory, and infectious disease applications. And it's well established that targeting a gene called FUT8 will produce afucosylated antibodies. So we looked at that. We had a very, very active guide pair for Cas-CLOVER, over 75% editing efficiency. So we had plenty of clones to pick from, and showing the frequency of the indels here, just another view of the size of the deletions that we're getting. You can see most of those deletions, where you see the count of cells on the y-axis and the size on the x, are right around 20 base pairs, but we did have some that were above 250 base pair deletions. We looked at doubling time. That's one area that FUT8 knockouts are supposedly get worse versus the parent, but we didn't. We had many clones that had acceptable doubling time of sub 22 hours. And then we wanted to look at the glutamine sensitivity again of these clones and make sure that they retained that, and they did retain that with good sensitivity over many, many clones that we isolated. So then we wanted to look at the productivity of some of our top clones. And we ran the same studies with a couple of our top clones shown here, with the antibody titer reaching 6 grams per liter. So very, very similar to our parent GS knockout. And then the cell-specific productivity was also very high. Of course, we wanted to look at the analytical to make sure that the fucose was gone, and we actually had a afucosylated antibody, so we did LCMS on the heavy chain and the intact molecule. I'm showing the heavy chain here, and you can see the control antibody is in blue, and then all of our clones have this shift down in size, where indicating that it's afucosylated. So that was confirmed. But the important thing is, do these cells produce antibodies that have an enhanced ADCC for therapeutic potency? And so we did a reporter assay, an ADCC reporter assay, where the dead cells emit light, they luciferase. And so on the y-axis, you've got the relative light units showing how effective this antibody is. And then you can see the antibody concentration on the y-axis. And we had a negative control, which didn't do anything. And then our regular trastuzumab, which was made in the GS knockout, had its potency profile. But then all three of clones that we tested had much higher potency, but also lower EC50, as you would expect. So the EC50 decreased 50 to almost 80%, representing an increase in potency between 2 and 4.8 versus the regular antibodies produced in the GS knockout. So shifting gears back to the stability of our systems, wanted to show how stable CHO pools using CleanCut and HarborIn can enable rapid transition to manufacturing. So this is stability data that we have with trastuzumab, and this is generation 0 through generation 60, and this is a pool, and antibody titer is rock solid. You get no changes. The cell-specific productivity is also stable with these pools. And then the copy number of the transgene is also stable in these pools over 60 generations. And you can see we get somewhere around 14 copies per cell. I also want to mention that the clones were also stable, but focusing on the pools, because I'm introducing our new partnership with a CDMO based in Sweden called NorthX. And what we're doing is we're offering cell line development services that feed directly into downstream CDMO services with NorthX. And we can greatly compress the timelines by first delivering the material on the bulk pools. It's extremely stable. This gets delivered to NorthX. They do preclinical or IND enabling material manufacturing. In some cases, you can imagine they could manufacture phase 1 clinical trial material. Process development is done there. At the same time, we are isolating the clones. So we're doing single cell cloning, characterizing those clones, doing the stability and titer, and then you get your clinical material for downstream, greatly reducing your timeline. And each month saves tons of money on your program. So this is the whole platform that we can now deliver, gene-optimized CHO cell line development to GMP production. So first, you start with the right CLD platform. I've talked a lot about our CLD platform, but I wanted to mention that we also have expert transposon vector engineering. So any molecule, any type of stable expression that you want to do, we have that. And then we have the ability to choose advanced CHO platforms, whichever one. You want afucosylated antibodies, non-afucosylated, and we're growing that platform. Then we've already qualified into NorthX, so they go right into process and analytics built around your molecule. So it's tailored upstream and downstream process optimization. I want to really emphasize the full analytical suite that they have and method development. So Usually this is done at a different CRO, but this is a very, very big bottleneck that they can solve. And then they have GMP execution. So 30 years of GMP biologics, many programs that have gone into the clinic. And then one of the best things is the integrated fill and finish and drug product analytics. So you don't have to go anywhere else. You just go right here and you get your clinical material ready to dose patients. So to close, our platforms are modular by design, powerful together. So you use Cas-CLOVER to edit your host. You can use the engineered host cell lines that we already made off the shelf for your production platforms. And then you've got the harbor-in transposase to integrate stably. And you can use any one of these components standalone, or you can combine them for a full workflow. Now with that, I'll take any questions. Here's our contacts, and there's a QR code if you want to access the research papers that I cited or book a consultation with us. I will be, and we will be at our booth 616 if you have any further questions as well. Thank you.
All right, guys, you know the drill. I'll start with my question. Thanks, Jack. That was really great. Very cool to see the top to bottom platform that you're coming up with. I do have a question about the stable pool generation and then making the IND enabling material. So my question is on the critical quality attributes. Are you working under the assumption that you will match your clones to the pool? Because I'll say that this is something that we think about, right? Can you use material earlier in the process? Yes, but it might come at the cost that your optimized protocols or standards that you set won't match the clone that you identify at the end, right? Like if you think about acidification, maybe your clone doesn't match the same species you set earlier. Have you thought about that or encountered that in your process here?
We have not encountered that. So my understanding is, you're really just trying to get material to make sure that it's not toxic, right? In the IND enabling study. And the pool is going to have cells that have some copy numbers that are different than others and maybe some variability in the glycosylation. But it's my understanding, and I am not an expert in this, but it's my understanding that the material from the pool can certainly be used for those early stage studies.
Thank you.
Hi, very, very nice talk. I did also have a question about stability studies. So the stability studies that you showed, did that include feudate knockout cells?
No, that's a good question. That stability study was with just the GS knockout.
Are there plans to do a feudate stability study?
We have not done one yet, but we are going to do that, yes.
Okay, great. Thank you. Be interesting to see.
Yeah.
Question about the stability for the harboring transposase. Looks as though the pool in 100 PDL is more stable than the original one. Is that molecular specific?
I'm sorry, what's the question?
So for your stability study for the pools, I saw the 100 PDL after you patch the cells many times. Looks the titer even higher than the original one. Is that molecular specific? It's common. Normally we say decrease. In your case, I say increase.
Yeah, I don't think that is something that we would see a lot, or I'm not exactly sure, but we wouldn't expect that. But there wasn't a whole lot of variability in there that I can recall. So I wouldn't count on more. I would basically just say this is stable and we're not going to see much less.
Okay.
In terms of productivity.
Yeah. Maybe that's molecular specific. I think that's not normal for me. Another question about the pool. So I say the pool, the titer, the high. So you use like separator for single cell cloning. How many clones do you scrape for from the pool? How many clones do you like evaluate in TPP or 24D12? So there's what's variability of those clones.
Yeah, so we don't have to screen that many. So we usually single cell clone 4 plates, and we usually screen about 100 clones. And they're all, there is a range somewhere between, like if the top clone is 7 grams per liter, we might see some clones that are 3-4 grams per liter. But most of the clones that come out of a good pool are good performers.
Yeah, so another question, just your top clones, what's the ratio of the titer with the pool? For example, you can have like 2 times or 3 times.
No, definitely not 3. What we see, not 3 times. More for us, what we see is more like half, like 1.5 times better with your best pool, because the pool is already very productive. So that's how we, so far.
Okay, thank you.
Sorry, I have a quick question. Thanks, nice presentation there. So, since this is an enhanced transposing system, my expectation is you're going to have something more like a narrow distribution, because if it's enhanced, then the population shouldn't be very variant, going from your bulk pools and even going to your clones. Is that what you're getting, or you're still having this very varied type of distribution when someone is using a wild type transposase?
We see not much variability. I mean, when we transfect the system, it delivers copies. It delivers about the same amount of copies in most cells, essentially.
Okay.
The beauty of it is, and there is some variability, but the beauty of it is it preferentially integrates at highly expressed, highly stable sites. So we actually don't need 30, 40, 50 copies of the gene of interest to get good titer. So I've seen some cells that have maybe 5 copies that have the same amount, the same titer levels as a 15 copy cell. But generally, we're looking at like 5 to 15 copies, and no bigger range than that.
Okay. Yeah, because one of the reasons I'm asking the question is, in as much as we want to narrow everything down, I also don't want to start screening so many clones as well. So if I have something that is pretty uniform coming from my stable bulk pool, going to my clone development, then I have lesser clones to deal with because I have so much I can work with. That's why I'm asking the question about how variable that stable bulk pool is, because I've seen quite a number of variability even with this transposites too.
We don't have to screen too many clones. Actually, we probably overdo it because we just want to be careful. But yeah, we see pretty uniform versus the pool.
Thank you.
Mm-hmm.
Let's thank Jack one more time and take our conversations offline. Thank you so much.
Thank you.
That was great.